or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. This episode is ad-free, and I rely on my audience to support my efforts. If you're getting value from this content, please consider becoming a paid subscriber to the Mind and Matter Substack. For just $5 a month, you'll get early access to episodes and other content, full episode transcripts, and I'll prioritize answering your questions in emails and the comments sections. You can also support me by checking out the links in the episode description to my affiliate partners who make various health and wellness products that I use and enjoy myself. You'll receive a discount and help support the podcast that way. And if you want the benefits of a paid subscription, but it's not in your budget, simply sign up for my free weekly newsletter, send me an email, and I'll give you a complimentary paid subscription. Word of mouth is the best way to help the podcast grow. So if you like what I'm doing, please share your favorite episodes with family and friends. The purpose of the podcast is to provide you with information obtained largely from primary sources, the people doing the research and producing new knowledge. This content is never meant to serve as medical advice. And with that, enjoy the episode. Tell everyone a little bit about who you are and what your lab studies.
Yeah, so I'm an Australian that's recently moved back from the US after about 8.5 years. I grew up in Sydney, so it's really exciting to be home and doing what I love. I generally consider myself a behavioral or cognitive neuroscientist. We work with rats, so that generally means you have to say behavioral instead of cognitive, though I don't necessarily agree with that. And what we're trying to do is to understand how different parts of the brain contribute to our understanding of our environment and help us learn about different contingencies in our environment so we can respond appropriately and get the things we need and avoid the things that we don't. We like using different techniques like optogenetics, which allows us to manipulate neuronal populations with light, with a lot of temporal precision, because we're really interested in the very specific ways in which different neuronal populations contribute to learning. And we also record neural activity using things like fiber photometry that allows us to record, for example, dopamine release with dopamine sensors and these sorts of things, again, in a way that relates to learning and specific things that are happening for the rat in their environment.
What is the difference between cognition and behavior? Let me just ask you that.
So I guess there's kind of a long-standing argument in the field as to whether rats are really capable of cognition. And a lot of people, often, I guess, human or primate researchers generally would consider rats not to have cognitive abilities, but rather exhibit behaviors that might mimic some of the behaviors that we mimic, but don't necessarily have the same cognitive processes that are underlying that.
Well, so what? So how exactly would you define cognition?
Sure. So I guess I would define cognition as the ability to relate new bits of information together to essentially make novel inferences about the state of the world in a way that hasn't necessarily been told to you, or a way that you haven't directly experienced in your environment before.
Mm-hmm. And so it's completely uncontroversial and obvious that primates like us have different mental and behavioral capabilities than rats and other rodents do. Can you can you steal me on the argument that rodents don't have true cognition before we maybe get you to provide counter arguments to that?
So I guess there's an argument as to whether they really have a prefrontal cortex. And so people would often think that a prefrontal cortex is what allows you to make these kind of novel inferences and perform sophisticated behaviors that might reach the benchmark of cognition. And so there's an argument that they don't have the same kind of prefrontal cortex that primates do.
I'm guessing you don't find that compelling.
I think that primate prefrontal cortex is definitely different from a rodent prefrontal cortex, but there's a lot of work that, we or the people that I've worked with have done that show prefrontal cortices in the rat do very similar things to what they do in the primate. So, for example, you can isolate, the Stroop effect or something like that in parts of the prefrontal cortex in the same way you can in humans.
Mm-hmm.
And I think that's really great evidence for inference and also for cognition, higher order cognition that's dictated by prefrontal cortices in similar ways across primates and rodents.
So what are some of the things that you guys study in the lab in rats that might be examples of cognitive behavior?
Yeah, sure. So one of the big ones is probably sensory preconditioning. And so that's essentially a task that's generally used to assess what we call model-based inference, which is just making an inference from information that you've learned about, but you haven't had to piece together in a novel way before. And what that involves is essentially, for example, teaching a rat, although this has been shown in primates and also in mice, teaching a rat, for example, that there's two neutral stimuli that are related. So, for example, one tone goes on for 10 seconds, and then an auditory click will come on for 10 seconds. And the animals learn that those two auditory stimuli are related in time, and so that they learn that they're essentially linked. But they don't mean anything to the rat. So the rat's not really doing anything about them. But you can reveal what they have learned about them by then presenting one of those cues, for example, the click with food separately. So on different days, they'll experience a click that comes on for 10 seconds and then the food is delivered. And they learn that there's a relationship between the click and the food across many days and many trials. And then what you can do is to present back that auditory tone that they first heard that predicted the click in a test by itself. And what they'll do is to exhibit an expectation for food, as though they expect food is about to be delivered. And we think that's due to a model-based inference, which is that they know the tone and the click are related together. They know that the click leads to food, and so they're making this novel link during that test where you're presenting the tone by itself for the first time, that it's also going to lead to food. And so that's what we essentially call sensory preconditioning and model-based inference.
I see. So you associate two arbitrary sensory stimuli together with a rat. So you present one, you present the other. They go together in time, but they don't mean anything. So the rat doesn't associate it with a shock or something bad happening or a treat or something good happening. And then later on, you pair one of those stimuli with a reward, and the rat automatically associates the other one with a reward.
Well, so there's a debate as to when it is that they associate the other cue with food. And so some people would argue that it's taking place during the learning about the click and food, and that's something called mediated conditioning. So the idea would be, okay, you've got this click that comes on, it makes me think of the tone that it was associated with, and therefore the tone and the food are going to become paired together because it's a mediated thing where you're just thinking about the tone at the time that the food comes. We have evidence that that's not the case. And so what we think is happening is that at what we call the probe test, where you present that tone for the first time, they're going, oh, hey, the tone, that leads to the click, and that click leads to food. So I think it's likely that this tone is going to lead to food. And so they go to the food port.
And actually, before we dive into some of this in a little bit more detail, maybe we should go over some more basic things. So, a lot of what you do would be considered reinforcement learning and associative learning and some of these concepts. What can you just kind of briefly describe for people what is reinforcement learning and what is association, and how are those things typically measured in a rodent?
Yeah, so we typically think of reinforcement learning as, you're learning about information in your environment because it's paired with something important. And that means that it's really important that you learn about it. And there's a whole bunch of rules that people have made in different models about how you associate those things together and how it weights up different things, like how far away a cue might be from the important thing like food, how salient the cue is, all those sorts of things. It's essentially reinforcement learning is essentially a lot of maths to describe the way in which we learn about things that are important to us. And an association would be a little bit different in that associative learning theory is really thinking about how we explicitly make associations between representations of things in our environment. So the difference, I guess, is in the representation in some ways, although it's not always as disparate. And that's thinking more about how you link things together. But there are also mathematical models of how you do that that can be very similar to reinforcement learning and computational reinforcement learning.
Mm-hmm. And so, like when we think about uh, studying rodents and studying learning and memory in rodents, um, oftentimes the first thing people learn about is Pavlov's dogs. So this would be a
Yeah.
simple form of association. Can you kind of give people a sense for like the different types of learning that we tend to see in rodents, that we tend to study, simple associations versus things that are contingent on behavior, and then maybe up to some more elaborate things that perhaps you or others have worked on?
Yeah, so I mean, really just like Pavlov's dog, where the dog is learning that there's a bell that is associated with food delivery and it has a conditioned response whereby they show salivating to the bell that's paired with food, even if you don't bring the food out. You can get rats to learn these sorts of associations between like a tone and food. So they'll exhibit an expectation for food when the tone comes on. They'll go and search for food when the tone comes on. They also can exhibit things like taste reactivity. So if you paired some kind of flavor or even an auditory stimulus with the tastant, you'll actually see that they exhibit the kind of oral facial activities that were associated with the tastant itself. So these are all kind of conditioned responses whereby cues are kind of cues in the environment being flavors or being tones or lights or something like that can take on some of the properties that the actual food itself has, which is Pavlovian conditioning.
And so on like your description of your research on your website, it says that you guys are looking at revisiting current theories that are often taken as dogma but may not be incomplete. Can you give us a sense for what some of those are? Where is sort of the field right now in terms of things that maybe people are taking for granted that we're starting to learn don't work the way we thought they did, given some of the detail we have now with new technologies that allow us to probe the brain more deeply?
Yeah, so I think obviously dopamine is a really great example of that, and we've done a lot of work with dopamine. I think there's lots of other areas of the brain where we thought we know what they do, but actually they're doing something more complicated. But dopamine is probably one of the most obvious ones where we've really changed the way that we're thinking about how that neurotransmitter and how dopamine neurons, so neurons that are releasing dopamine, are contributing to learning and cognition.
Mhm. So I think some of the stereotypes that are out there, especially in the wider culture, but even among scientists to some extent, I think dopamine is strongly associated with pleasure, with hedonic value, with drug addiction, and things like that. Over the past, I don't know, 5, 10 years or so, since you've been in this field, how has thinking among neuroscientists who study dopamine and related things, how has it started to change? And what are some of the misconceptions about it that are out there?
Yeah, so I think it's still hotly debated as to what dopamine does. And I think more and more we're understanding that dopamine does a lot of different things. One of the ways in which we've contributed to the literature is that we've shown that dopamine can help you learn associations between things rather than just serving as this chemical that might endow food or cues paired with food with a kind of hedonic pleasure or something like that. So one of our papers, for example, showed that we could drive learning in sensory preconditioning, which is essentially learning between two neutral sounds in the environment by stimulating dopamine optogenetically as a prediction error, which is how dopamine neurons usually fire during learning. And we showed that you could essentially get animals to learn about the associations between those neutral stimuli in a way that couldn't be understood by just assigning value or pleasure or something to those cues. So that suggested that dopamine contributes in more complex ways to reinforcement learning and really is helping us to understand our environment and the complexities within it over and above a role for it in assigning pleasure or some kind of hedonic valence towards different stimuli or food itself.
Mhm. And I mean, does it even make sense to you that we should be trying to talk about, on the one hand, there's a challenge here from like an educational perspective. If you're a scientist studying this stuff, at some point, the information has to get out to the wider world, and we have to obviously use words to do that and simplify things. But on the other hand, a lot of misconceptions can pop up, and we can sort of get things very wrong if we're not careful with how we speak about some of these things. With dopamine being equated with things like pleasure and things like that, you often hear that about other neurotransmitters as well. People will talk about serotonin as being for mood or emotion, as norepinephrine as being, for attention or about attention or something. Is it useful to equate neurotransmitters and molecules like this with individual concepts as long as we're careful, or is it just fundamentally too simplistic and we should avoid thinking like that?
I think that's a really interesting question, and I don't really know what the answer is. I mean, I think in a lot of ways it was really smart of people that came before to be talking about dopamine as being involved in reward or hedonic pleasure, because there was evidence that, dopamine neurons are just firing to things in the environment that predict something good, which is explicitly food or reward. And so in some ways that was really a useful way to communicate to, the general audience about what it is that dopamine is doing. I don't think that it now captures what we understand dopamine to be doing. And I guess now we have to think about a way to communicate more effectively in simple terms what it is that dopamine might be doing to help us learn about our environment and represent the different associations within that.
Mm-hmm. I often hear people now talk about dopamine as being involved in motivation in a more general way than just being narrowly focused on pleasure and reward. Would you say that's accurate? Slash, how would you, given the state of the art today, how would you talk about our understanding of dopamine and what it does?
Yeah, I mean, I think dopamine does a lot of different things. I'm just going to close the window over there for a second.
Oh, yeah, no worries. One moment, folks.
Lots of kids going to school. Yeah, so I think dopamine is definitely involved in motivation, and the magnitude of the dopamine response is going to influence how much, for example, a rat responds to a cue that's paired with food. A lot of the work that we've done, we actually have a paper that's coming out soon, shows that you can dissociate the magnitude of the dopamine response in somewhere like the nucleus accumbens from responding itself. So I think dopamine is reflecting the extent of learnt associations or the magnitude to which you've associated, for example, a cue that might be paired with food, and that influences responding. But what we're finding is that you can dissociate that from the actual response itself. But we know that dopamine, and particularly in the striatum, is really important for how you respond and when you respond, and whether that is the cause of the response or associated with the response itself. I think we still haven't really dissociated.
What so when we talk about dopamine, we often talk about people will say the reward circuitry, the mesolimbic dopamine reward system. They'll talk about places like the nucleus accumbens that you just mentioned, the ventral tegmental area, the synapses where dopamine is released that are really important for things like reward learning, really important for things like addiction, and other things. But is that the whole story, or are we sort of learning and piecing together a wider and more elaborate circuitry that involves dopamine? And if so, what are some of the maybe key circuits that you've been working on or that we've discovered more recently that connect to but aren't just that classic VTA to accumbens connection?
That's a great question. And so this is what I'm really excited about in our lab and like generally in the field. So I think now that we're understanding that dopamine's doing something more complicated, we now know that the prediction error signals and other type of signals that are coming from dopamine neurons in the ventral tegmental area, which is kind of the source of these learning signals, right, is going to be doing something really different in a lot of different circuits, because dopamine neurons project all throughout the brain, right? And so one of the things we've been doing, for example, like many in the field, is to try to understand how these different circuits are contributing in different ways to cognition. And I kind of thought this was, not a boring question, but I just thought that we'd be recording prediction errors all over the brain in different
Mm.
circuits. And then you kind of figure out how it is that they contribute in different ways to learning, which is probably like very similar to what we think the brain region that's receiving the signal is contributing to cognition, right? But actually, what we're seeing is something that's really different in our lab, and we're seeing really different kinds of dopamine release and dopamine signals in different circuits that really don't have a lot in common with one another. And so it doesn't just look like these areas of the brain are receiving prediction errors in the way that we think about a phasic prediction error that comes from VTA dopamine neurons and gets sent to places like nucleus accumbens. The signal looks really different. And I think that's really exciting because it's telling us a lot more about how it is that dopamine is contributing to cognition, and really, I think, showing that dopamine is extremely influential in the way in which we're learning about our environment by dictating what is learnt downstream, is the way that I'm thinking about it.
I see. So it kind of sounds to me like, so there's this area called the VTA, the ventral tegmental area, in the hindbrain, basically, deep at the bottom, sort of primitive part of the brain, and it's famous for, it has the neurons that release dopamine. They release it in the nucleus accumbens and the so-called classic reward circuitry, but they project all over the brain. And they're also famous for doing something called a reward prediction error, which is, I don't know how you would say it. So sort of like a kind of subtraction that neurons can do that scales with how surprising an outcome is. And I guess what you were saying, if I heard you correctly, is we know that there's this reward prediction error coming from these dopamine neurons in this classic reward circuitry synapse. And the VTA projects there and a bunch of other places. So it's probably just sending the same type of prediction error everywhere else. But it sounds like we're starting to discover that's not the case. It projects to other places, and there's other types of computations probably happening.
Yeah, exactly. It looks really different. And we do know that there's a lot of heterogeneity in dopamine neurons themselves in the VTA in the
Mm.
way that they respond. And I think it's circuit defined, which many other people in the field have shown in different ways. And now we're starting to understand the extent of that. So the extent of heterogeneity in the response from VTA dopamine neurons that might be kind of dictating what is learned downstream in many different regions. But even in the nucleus accumbens itself, where we think of this as really receiving and being a proxy for the reward prediction error that you were talking about, we've shown now that in the nucleus accumbens core, for example, that's a particular type of learning that's being reflected there, which is not the whole error, right? It's not the whole component of what is learnt about cues and rewards. So even that is this much more specific thing than what we've previously thought about. And I think that is really illustrative of what we're going to find downstream in other regions is they're doing something much more specific than what we thought they were in terms of how dopamine's contributing to that sort of learning.
So these dopamine neurons that live, that where their cell bodies are in the ventral tegmental area in the hindbrain, they project to multiple areas. Can you give us a sense for where? Is it truly all over the brain? Is it several regions? Can you give us a picture of that?
Yeah, I mean it's a lot of different regions, right? So it's ventral striatum, which is nucleus accumbens, it's dorsal striatum, dorsal lateral striatum, and dorsal medial striatum. So basically the whole striatum, which is we often think of as being important for responding and things like that. It's also areas we've revealed like the lateral hypothalamus. So we've characterized a novel circuit there where dopamine neurons are projecting to the lateral hypothalamus to facilitate learning in unique ways. Also, recently, we're looking into projections to the ventral hippocampus as well. But dopamine neurons also project to the dorsal hippocampus, and the hippocampus is often thought to be, really important for cognition and forming associations between things that are related in your environment. So that's a really important projection because it really is showcasing the complexity of what dopamine can do. Dopamine also projects, of course, to prefrontal cortex, many different regions in prefrontal cortex, and what it's doing there has been known, it's been known for a long time. It's doing something influential, and I think it will be really interesting to understand exactly what it is that those terminals are doing.
Hmm. Yeah, I want to dig into that a little bit more. But earlier you mentioned something called model-based learning. Can you contrast that with model-free learning? I know this is something that you've worked on, but what are those two things, and how do you study those in rats, for example?
Yeah, so model-based learning is essentially where you are learning about how things are related in your environment in a way that allows you to make novel inferences when you're faced with something new, right? And the way in which we test that in rats, for example, is in sensory preconditioning, where we teach them that two neutral cues are related, and then we teach them that one of those cues leads to food, and the animal will make the novel-based inference, model-based inference that the other cue that hasn't been paired with food is likely to lead to food. And that would be an example of a model-based inference because they're using information that they've learned in the past to predict something new. Model-free learning is where we're essentially assigning a cache value to cues or other things in the environment, like context, that have been associated with something of value, like food reward. And essentially, that cache value is thought to have backpropagated from the food itself. So there's a kind of number that's associated with that food, like a 7 out of 10, and that 7 out of 10 attaches to the cue. So it's not teaching you about the relationship between the cue and the food, right? It's just making that cue valuable in and of itself so that you want it, but you don't necessarily know why.
Hmm. So, I mean, off the top of my head, if to give people an intuitive sense for this, if we were thinking about something like a food reward or an addictive drug, something naturally rewarding that you really want, a tasty dessert or something. If you're sort of thinking about that in a model-free way, I don't even know if I should say thinking there. If you're looking at that in a model-free way,
Yeah.
that would be like if someone had like a compulsive addiction or orientation to this, like, okay, I want that piece of cake right now, and it's just sort of like, boom, I see it, I want it, I know it's tasty. Whereas if you had a model-based way of learning about that, that would be a situation where you're thinking, all right, there's a piece of chocolate cake there, but I just ate a little while ago. I'm trying to lose weight. Do I really want to eat the tasty thing, or do I want to hit my weight goal? The more deliberative, cognitive way of making a decision.
Yeah, I think that's a really good way of describing it. And that's often how we describe it in our papers as we kind of attach it to examples in our environment. The idea is that model-free kind of agent, as we call them, a model-free person or rat or whatever, essentially isn't thinking about their actions. They're just kind of attracted to these things and they're not really thinking about the consequences of that. And often it's thought that in addiction, these processes are heightened in some way so that people are not able to kind of avoid drug paired stimuli and stuff like that because it has this very high model-free cache value. Whereas if you're a model-based agent and you're performing more on the basis of model-based associations and inference, then you're going to be able to say, well, no, I really don't want to do that because it's going to have negative implications on my life. And so I stop.
Mm. I mean, this immediately makes me think about things like drug addiction and how they tie into development. So you could imagine if you form associations between a stimulus and a strong natural reward early in development before someone's even gotten to the point where they have model-based learning, you could imagine that would have very different consequences from introducing a reward later in life. So maybe there's an interaction between the strength of reward and someone's ability to react to it non-compulsively based on where in their cognitive development those associations were formed.
Yeah, I mean, I think that's a really interesting way to think about it. I haven't really thought about it like that before. I guess in part because I don't necessarily buy into the very strict model-free, model-based argument of addiction,
Mm.
I guess. But I think that would be a really interesting test of that. So we think, that rats develop model-based learning and that primates develop model-based learning kind of around adolescence when they develop a prefrontal cortex, right, properly, and that whole circuitry becomes defined. So if they experience some kind of drug cue association, would that mean that they are then more likely to learn about those types of associations in a model-free way? Yeah.
Yeah, I guess partly what I was thinking there too, and I'm just thinking off the cuff here. When you see something like a compulsion develop, say like a drug addiction, is that because there is this just super strong sensory reward association. It just sort of is louder than any competing association? Or is it somehow like preventing model based learning from even happening?
Yeah, I think that's really interesting. So the, traditional view on this is that we have model free and model based processes that are constantly going on in parallel, right?
Yeah.
And for some things, they're weighted differently. So, for example, when you start driving, like you're going to be acting in this very model based way because you need to think about everything that you're doing and the consequences of that. But later on, you become really efficient and really good at this. So you're going to become more model free and develop the kind of habit, which is what we call essentially a well established model free association. And the idea is that in addiction, taking the drug itself is essentially weighting your brain more towards the model-free learning and the model-free behavior, which means that these associations with reward and the value that these cues can have are having more control over your behavior than a more deliberative process that allows you to think about the consequences of your actions. The problem is it's getting more complicated. There's always these really nice theories that have a lot of evidence for them, and they're kind of easy to convey in a way, like the kind of action habit drug addiction argument, which is the same as the model-free, model-based drug addiction argument. But there's now a lot of evidence that a lot of drug-directed behaviors are actually the result of a model-based agent, so a model-based process. And a lot of these cues that are associated with drugs are essentially able to control behavior in a model-based way, and that still has this huge effect on how much they're going to seek out the drug and how much they're able to stop trying to seek out that drug. So it's
Yeah.
getting more complicated.
Yeah. And I think, if I just think about this off the cuff, you could imagine, if someone's going for the reward, let's just call it a drug reward. There's two ways, at least, that they could take the reward. One could just be, they don't
Yeah.
even think about it. Just boom, boom, association, take it. It's just completely
Yeah.
like zombie behavior. But the other one could be, they think about it. They go, I don't know if I should take it because there's all these negative consequences. And yet they're still compelled to take it after deliberation.
Yeah.
We've probably all been in those situations, right? Where we're thinking about
Yeah.
doing something and we do think about it. And yet, we go with
Exactly.
the reward anyway. And it sounds like you're sort of saying the second thing has been observed.
Yeah, I think that's a really, really great way of describing it, right? That there are two sort of ways that we can go about this. And it does look more and more as though people, for example, that are struggling to stop taking drugs are thinking about it and they are
Yeah.
thinking about the consequences, but something else wins out, right? The
Yeah.
power of the Q drug association wins out. And I did my postdoc at the National Institute on Drug Abuse, where people are really thinking about and have been thinking about this in those terms for a long time. And one of the examples that they often bring up, or that I often heard when I was training there, is, people that have severe drug addictions, right? That are forgoing jobs and family and all these sorts of things to keep taking a drug. They go to really complicated lengths to try to get the drug.
Mm-hmm.
Right? There are crazy stories of things that people are doing to try to get hold
Yeah.
of these drugs. And that
Yeah.
doesn't
Meaning,
sound like,
yeah. Meaning there's elaborate
yeah,
planning and a lot of cognition going into it. It's not just simplistic, zombie
exactly.
behavior.
Yeah, exactly. Exactly. And when you think about it, in the way that addiction looks like in the real world, it doesn't sound like people are just like picking something up and taking it, right? There are all these things and all these plans that are going into it to be able to procure that drug. And it's not easy in our environment,
Yeah.
which I thought was a really interesting way to think about it. And our data are consistent with that.
So stepping back for just a second, I had a discussion with someone about a different subject the other day. We were talking about animal models and how well like animal models of depression actually model human depression and the translatability there. And the opinion of that person was basically like, well, we've had so many animal models of depression, rodent models of depression for so long that, we should be on like ninth generation SSRIs by now if they were modeling depression in humans very well. So his opinion was, rodent models of depression, are not modeling human depression as well as we would like. I'll put it conservatively like that. And obviously, in the literature, people are careful to say things like anxiety-like behavior, depressive-like behavior, and in the addiction and reward, there's an analogous thing I could ask about addiction reward and how well rodent models capture human addiction. But just at a basic level, how well do some of these rodent models actually capture the phenomenon that we're interested in from a sort of human translational perspective?
Yeah, I mean, I think that's a really complicated question with a complicated answer. I'm not sure I'm the best person in the field to answer it. I guess the way that I would think about it is that there are some things the models are able to capture and some things they're not. So, for example, this change in goal-directed habitual behavior, which is like model-based and model-free behavior, seems to come out in humans and rodents that have been exposed to drugs. So there are definitely changes in cognition that look really similar in a rodent model of addiction to humans that have addictions to different substances, right? And I think that's probably our best evidence for translation. In terms of how an animal responds for drug, actually rats often don't really like drugs. And there's usually just a subset of rats, of course, that will go on to exhibit what looks like addictive kind of behavior. And people have captured this in a number of different ways. I think it's like, 15% of rats or something. And people say that actually that's quite like humans, right?
Yeah, I've talked to Christian Lucier, for example, on the podcast, and in his, I mean, this is a very reproducible result. I'm just going to be specific. It's rats and cocaine I'm talking about here. I don't know, but I assume that this generalizes to some extent. Basically, roughly 30% of rats will become addicted to cocaine if you give it to them over and over again, and they're in a not too enriched environment. And that apparently is like the number for humans as well. It's actually remarkably similar, despite the fact that, rats and mice are inbred populations and we're completely different animals, et cetera, et cetera. And so with that in mind, so when you say rats don't really like drugs, do you mean that only a minority will proceed to compulsive drug seeking? Or do you mean that like they often don't find drugs rewarding that we do?
I think it's a little bit of both. You definitely have to work harder to get rats to take drugs, right? And I don't know whether that's because, obviously we can't do this with humans, and maybe humans, 85% of humans would show the same kind of thing. But a lot of the time, people are doing things in the lab with rats to get them to press a lever because they won't press as they will for food or something like that. And it is definitely... Yeah, sorry.
I was just going to say, like, so I think there's a bit of a misconception out there. People probably think it's probably pretty easy that if you just put cocaine in there, they'll just start pressing it forever and ever automatically. Yeah. That's not the case.
No. It's quite hard to get them to do it. There's a lot of kind of ways that you can get rats to behave, right? And the people in the addiction field often use those to get them to start taking the drug, right? Because it's quite hard. But then, as you're saying, Christian said, and lots of the people in the field have found that there are about 15% or something of rats that really like it and will very happily take it and will forgo food for the drug and will also, undergo punishment. So get shocked sometimes for pressing to take the drug and things like that. So maybe that is actually a good model for what humans are also like. But yeah.
So VTA, it's where these dopamine neurons live, that we've talked about a little bit. They project famously to the nucleus accumbens. That's where we see this reward prediction error signal that's been fairly well studied. That's really important for reward learning. And it's part of the classic sort of reward circuitry, the mesolimbic dopamine system. But as you mentioned, these VTA dopamine neurons project to all sorts of places. One of those places is the lateral hypothalamus, and that's a place that you've worked on. So before we get to some of the specifics there in terms of the dopamine circuit, can you give everyone just a brief, concise crash course on the hypothalamus and the lateral hypothalamus? How do we normally see these things talked about in textbooks, say?
Yeah, so the lateral hypothalamus is my favorite topic to talk about. Lots of people like talking about dopamine, and people come to the lab and they want to do stuff with dopamine, but the lateral hypothalamus is like really where my jam is at right now. Essentially, we thought that the lateral hypothalamus is the feeding center of the brain. So the idea is if I was to turn on your lateral hypothalamus right now, you'd like start eating whatever is in front of you and maybe even start gnawing on your microphone or something like that. It's like thought to just drive this innate motivation to consume food and approach food-like substances. But during my postdoc, I got really interested in how a role for lateral hypothalamus in approaching food might actually reflect a really basic role for this area in learning about the predictors of food. So, for us, feeding is all about learning, right? We're not born with an innate understanding of what food is and how it reduces our hunger. In the same way, we actually have to learn that water is rewarding and it reduces our thirst. So we're not actually born with that. It's all about learning. And so we ran a series of experiments that tried to understand how it is that the lateral hypothalamus is important for learning about the predictors of food. For example, even the sight or smell or taste of food as being predictive of the reduction in your hunger and the intake of calories. And that sort of put us on a trajectory of a bunch of different experiments that revealed some really surprising things about what this area is doing. So we've shown now that the lateral hypothalamus is really critical for learning about cues in the environment that are really close to food. So it really wants to learn about anything that is close to food and the association between those things. But actually, it really wants to stop you learning about anything that's not close to food. So what we found is that it seems to be opposing learning about, for example, neutral cues. We do that using things like sensory preconditioning. It seems to try to stop you learning about the association between those neutral things. And the model that we've now developed is that the lateral hypothalamus is really important for kind of getting you to learn about everything that's proximal to what you need, which includes food, and to ignore everything else that isn't close to food, right? And so we think that this is an area of the brain that's essentially trying to bias you to learn about primary reinforcement.
I see.
So that you survive.
I see. And this is probably why I'm going to sort of betray my background a little bit. This is probably why classically, decades ago, the lateral hypothalamus was always associated with things like the orienting response. Which way, what is the animal paying attention to? How is it pointing at sensory organs? It sounds like behaviorally what's been known classically about this area fits with what you just described.
Yeah, I'm actually not aware of those studies. I would love to see studies looking at lateral hypothalamus and orienting to different cues that are paired with food and stuff. That doesn't surprise me. That fits with that. I think that's great. Yeah.
Mm-hmm. And so before we dig into this a little bit more, is it fair to say, so you said, we're not born actually knowing that, water is going to quench our thirst or that specific things are going to quench our hunger. Is it fair to say that animals are born with hunger and with thirst? They're born with the affective states that upset us, when we're deprived of those things. But we have to actually learn through association or through other means, what are the stimuli in the environment that will actually get rid of those feelings that we're born with.
Exactly. So that's why we have all these innate things in our body that make us do stuff when we're born, like go towards our mum's breast, right? That's so that we learn that association. So you don't have it innately. What you have is this response to do that with your mother when you're born. And that's how you learn about those associations to start with.
Yeah. And then so the hypothalamus generally, so it's hypothalamus below the thalamus, it's sort of at the top of the brainstem. Classically, is it fair to classically, historically, people would normally talk about the hypothalamus as being a quote unquote primitive or ancient part of the brain. It's a neuroendocrine organ, and it's involved in relatively simple things. It's involved in automatic or innate behaviors. It's not involved or thought to be involved in thinking stuff or cognitive stuff. Is that an accurate way of describing how people have historically thought about it? And is it sounds like, in fact, what you're discovering is it is doing some more sophisticated things.
Yeah, exactly. That's exactly the case. So that's what we always thought hypothalamus was doing. And this kind of circles back to this idea about prefrontal cortex and that being important for cognition, and how rats can't have cognition in the same way because their prefrontal cortex is a bit different. And prefrontal cortex is doing all the thinking, right? It's this real top-down approach to the way that we learn about our environment and the way that we behave. But actually, even what these subcortical regions are doing, like VTA, like the lateral hypothalamus, it's really complicated, and it is cognition. Yeah.
Yeah, I mean, it's amazing to me that I could understand historically why so many people started to think in terms of like, okay, lower brain regions,
Yeah.
so-called, they're not doing cognitive thinking stuff. Neocortex is, but that just seems to have completely persisted over time, even though we know that many subcortical regions are doing quote unquote cognitive things. That's quite clear. And we know that there's many animals that don't have homologous structures at all, and they're doing those things as well.
Yeah. Yeah, no, I agree with that.
So, can you describe some of these experiments that you've done in a little bit more detail with respect to this dopamine lateral hypothalamus circuit? What does the circuit look like? Are there dopaminergic projections going to the lateral hypothalamus? Is it bidirectional? What's sort of the structure of this circuit?
Yeah, so it's a bidirectional circuit. So we've known for a long time that lateral hypothalamus provides input to dopamine neurons in the VTA. And we've done some experiments to show that that's really critical for the learning phenomenon that we find with lateral hypothalamus. Now we've revealed that VTA dopamine neurons also project to the lateral hypothalamus. And they have terminals there, and they're regulating learning in the lateral hypothalamus as well. So the way that we think about it is this bidirectional learning circuit where VTA dopamine neurons are sending their teaching signals to the lateral hypothalamus so that the lateral hypothalamus biases learning towards cues that are proximal to food, and it then relays that expectation back to VTA dopamine neurons so that VTA dopamine neurons can do their subtraction of the prediction error that essentially allows it to calibrate how surprising something is and whether it's going to send out a teaching signal to keep getting you to learn or whether learning is done.
I see. And so is this projection doing something like the classic reward prediction error computation? Have you recorded that?
Yeah, so, well, we've recorded dopamine release in the lateral hypothalamus across learning. We've also optogenetically inhibited VTA dopamine terminals in the lateral hypothalamus during learning. And we find that the optogenetic experiments reveal that lateral hypothalamus is learning associations between cues and rewards, and this is facilitated by a dopamine error. We don't know exactly what that dopamine error looks like yet, but what we've been doing is to record dopamine release during cue reward learning in the lateral hypothalamus, and what we see is a really unique signature of dopamine release that doesn't look like what we expected it to, and essentially it ramps up as you get closer to the reward. So a 10-second cue comes on, the food comes at the end of that, and as the animals learn, you see this increase in dopamine release that ramps up to reward delivery. And this is consistent with an idea that this area of the brain is trying to get you to learn about things that are close to food.
Yeah.
This neural signature is consistent with that. And so we're really excited about that.
Yeah. So the level of activity is proportional to proximity to the reward.
Yeah, that's what it looks like. And that's what we want to understand more. So we're developing different tasks so that we can record this activity with cues that are proximal to reward, but also cues that are further away from reward. And what does lateral hypothalamus do with those cues? Because we know it's opposing learning about those. But what is the neural signature that correlates with that?
Yeah. And what does the local circuitry in the lateral hypothalamus look like?
Yeah, so it's really interesting. There was a paper that came out, I think, maybe a year or two ago, not so long ago, that shows that there's ultra-sparse connectivity within the lateral hypothalamus itself. So what that means is that neurons aren't really talking to one another, which is kind of surprising because usually throughout the brain, you have all this kind of local communication that's happening between neurons. And that's not what happens in the lateral hypothalamus. It's mainly receiving input, sending output.
And but like, what kind of neurons live there? Are these mostly glutamatergic? What are they?
All different kinds. It's one of the most diverse area of the brain. So we study particularly the GABA neurons. They release GABA, and we think they're really important for learning. They're also the main projection site, the projection neurons to ventral tegmental area, which is part of the reason we think that they're really important for learning. But you also have glutamatergic neurons, and they seem to be doing something different from the GABA neurons. You also have MCH releasing neurons. You also have orexin releasing neurons, and a whole bunch of other types of neurons that are in there that are regulating behavior in different ways.
And so you said earlier, I believe, that the lateral hypothalamus seems to have specificity for food rewards. Is it truly food rewards, or is it any natural reward, or do we just not know yet?
That's a great question. So other people have shown that it's also other types of rewards. So we know, for example, that drug rewards recruit the lateral hypothalamus. And one of my friends and colleagues, Nathan Marchant, has just published a paper a few months ago showing that cue alcohol memories are stored in the lateral hypothalamus and learnt in the lateral hypothalamus. Kay Ty's group have shown that the lateral hypothalamus is important for social interaction, and particularly the GABA neurons as well. So it's definitely a number of different rewards. We also have a kind of crazy result, which is one of the main things we're doing in the lab that show that the lateral hypothalamus can also be recruited to learn about fearful memories, which is really interesting. And the way in which it seems to be recruited to do that is that if rats have recently had an experience in learning about cues and food, then these neurons, specifically the GABA neurons in lateral hypothalamus, will become recruited to learn about the fear memory. And when that happens, it seems that the traditional fear circuit is no longer necessary. So it seems that lateral hypothalamus in some way is primed by the learning about cues and food to essentially encode something it wouldn't usually encode in the form of a fear memory, and it seems to take it away from the traditional fear circuit. And this is something we're going to publish in the next couple of months.
I see. So roughly speaking, if I'm hearing you correctly, traditional fear circuit elsewhere in the brain, we don't need to get into that, other parts of the amygdala and things, that would normally be where we would find a fear memory, like an association between a shock and a tone stored.
Yeah.
But if you prime the lateral hypothalamus with having a food reward given to the animal, that circuit can sort of take over the role of the other circuit for the fear memory.
Exactly. And it's specifically learning about food, specifically learning about associations that are proximal to food that seems to prime the lateral hypothalamus to do this.
What happens if you just damage the lateral hypothalamus, either completely egregiously, or and I don't know if this has been done, but with some specificity, like maybe just taking out certain types of neurons?
Yeah, so you would find a reduction in food intake, and if you lesion the lateral hypothalamus, they would starve and die. So it really is important for feeding, right? Because it's important to learn about what is food in the environment, essentially, is what we think. If you do it more specifically, so you can ablate the GABA neurons, and you'd also find reductions in food, but no effects on locomotor activity or anything like that.
I see. So if you damage this area, more or less, the animals would look fine to the naked eye. They'd be, sniffing and walking around in their cage and stuff.
Yeah.
But they just wouldn't be able to get to the food or figure out how to get it.
Yeah. Yeah, exactly. And we use things like optogenetics. So we can see how it is they respond very specifically to different things in their environment. And so we can optogenetically inactivate lateral hypothalamus just for a period of 10 seconds. It won't affect their locomotion in the cage, but they will specifically stop showing food-directed behavior when you present a cue that's paired with food and that they've learned is paired with food.
And so what else do we know about the lateral hypothalamus that in terms of like what it's doing generally speaking? So for example, like the one other thing that would come to mind for me is you mentioned that there's these things called orexin neurons that live there, and they're important for sleep and wakefulness. So do animals, when you destroy this area, do they show any other, if they have normal locomotor activity, do they have normal sleep and wake activity?
I imagine they wouldn't. I don't know the data on that.
Okay.
But I think that's really interesting. And I think we think the orexin neurons, as well as doing what you were talking about, like wakefulness and arousal, is also really important for learning too, and for processing rewards as well. So it's really interesting what these different populations are doing.
Interesting. So what are some of the things that you guys are working on today? Some of the questions that you're asking?
Yeah, so one of the major questions that we're asking is how can we understand the way in which the lateral hypothalamus might be dysfunctional in different models of psychological disorders? So, in particular, we're really interested in this bias that the lateral hypothalamus has towards learning about proximal cues that are near to rewards and stopping learning about things that are further away, like neutral stimuli. And the reason we're really interested in this bias is, for example, in drug addiction, you have this bias towards cues that are close to drugs and also actually other rewards, right? And so we think that that might be consistent with an idea that this bias is upregulated via an upregulation of activity in the lateral hypothalamus that might be producing this bias towards drug cues and other reward cues. And we think that this is important and also mirrors the literature because in rodent models of addiction, you find that those rodents learn less about neutral cues and things like that. So they're really showing more of a bias towards rewards and drug cues and things like that. On the other side of things, we know that something like schizophrenia is characterized by too much learning about neutral cues and things that aren't predictive of the things that are important to you. And people with schizophrenia also show a deficit in learning about the proximal predictors of reward, right? And so we think in drug addiction, lateral hypothalamus might be upregulated to produce an exacerbated bias towards reward cues. And in schizophrenia, this might be the opposite. It might be downregulated to produce an increase in learning about neutral cues and a decrease in learning about cues that are proximal to things like reward.
I
And
see.
we think this is a really critical balance in cognition that the lateral hypothalamus is doing that could kind of unlock a lot of our understanding of these disorders.
Yeah, I mean, yeah, it's interesting, like this balance between hyper focus and the spotlight of your attention versus
Mm,
seeing the wider picture. I never really thought of it this way. So a schizophrenic is sort of like they are literally learning things that aren't there to be learned. They're projecting sort of value and meaning onto things that are completely neutral in the environment.
yeah.
And what you're saying, and I didn't know this before, is they sort of are unable to learn about the things that they should be learning about right in front of their face.
Yes. Yeah. Yeah, exactly. And actually, there's no other area of the brain where you inactivate and it does those two things.
Yeah.
So you can inactivate, for example, the orbital frontal cortex. You'll get less learning about neutral cues, but you don't see the opposite effect on reward learning,
Yeah. Yeah.
right? And you can inactivate the basolateral amygdala and you'll get less learning about shocks and rewards, but you won't enhance learning about neutral cues. So this is the one region of the brain that we understand so far to be mediating this bias that
Mm-hmm.
is really critical
Mm-hmm.
to cognition.
Yeah. And so given, and I don't know if people have done this work or you're thinking about this at all, but it strikes me that, so this is an area of the hypothalamus. We know that hypothalamus is typically thought of as this sort of innate behavior, primitive neuroendocrine organ, whatever you want to call it. And yet the lateral hypothalamus is doing all these impressive cognition-like things. When I think about something like feeding and cognition, and I think about where that can go awry, I immediately think of eating disorders like anorexia. And obviously those are involved in food reward learning and associations, but they're also like, I guess you would call them cognitive. Like someone with anorexia has to make a story in their head about food that's pretty sophisticated before that can turn into full-blown anorexia. Is there anyone working on this, or is there anything known about this region with respect to something like anorexia?
Yeah, definitely. I think there are a lot of people that are working on stuff like that. So we have, for example, some Australian researchers, Claire Foldy and Zane Andrews, who are working on this in a model of anorexia, which I think is really interesting. But yeah, I wouldn't know the exact specifics of how they think lateral hypothalamus contributes to that, although, I could make some speculations.
Mm-hmm. And what were you doing? So before you started your lab, what was sort of your trajectory when you did your PhD and your postdoc? What got you to this field?
Yeah. So so in Australia, what we do is we do our undergraduate degree and then we do a fourth year where we can do a research project.
Mm-hmm.
And it's called an honors year. It's essentially the same as a master's elsewhere in the world, but we tend to do it in our fourth year as part of our undergraduate degree. And so I did that. And I just fell in love with neuroscience and research, kind of surprisingly. I didn't necessarily expect that. And then I went on to do a PhD, where I also just knew that I wanted to continue to be able to do research and to have a lab and all those sorts of things.
In
And then,
Australia?
yeah, I was in Australia where I did my PhD, at the University of New South Wales. And then I got an opportunity, luckily enough, to go and work with Jeff Schoenbaum and Yael Niv, over at the National Institute on Drug Abuse in Baltimore and Princeton University. It was like a joint postdoc between those two researchers, which was amazing.
Hmm. And then, so you recently moved back to Australia. How is the research culture and environment different or similar between the US and Australia?
Yeah, so I mean, I started my lab at UCLA, and that was a really amazing place to start my lab. I had a huge amount of resources that really allowed me to develop a lab that I thought was doing really cool stuff. It was kind of sad to leave UCLA. I think I always sort of wanted to go home. I felt really guilty leaving, but I really felt that I needed to prioritize my family and being closer to there. So I decided to leave UCLA and come to the University of Sydney. I think we definitely have less resources than what US institutes like UCLA have, but it's also less expensive to run my lab here. So there are a lot of things that
In what
we
ways?
already have at the university. Sorry.
In what
Yeah.
ways is it less expensive?
Yeah. So for example, like there's a lot of support for PhD students. So I don't pay to
Mm.
have PhD students in the lab. I don't need to pay their salary and things like that. At UCLA, I was topping up PhD salaries because they're not paid enough, right? And hopefully that's changing with the strikes and everything like that. But in Australia, they're fully funded positions. So we don't have to
From the
pay
universities
their
themselves.
salary. Yeah.
Yeah.
Well, from the government and the university, and there's kind of, yeah, a bit of both, but there's a
Okay.
lot more government support for universities here.
Yeah, I think a lot of people probably don't realize that about the US. Like the PhD. So obviously you can get fellowships and things that make you cheaper, that basically pay for your time as a PhD student. But by
Yeah.
default, right, your lab, the person who's running your lab, the PI, the professor,
Mhm.
they are getting grants that come into the university. The university takes quite a lot of that money, and then it's just gone. And then from the rest of the money, part of what you have to do is pay, pay the stipend or pay the effectively the salary of the people working in your lab. It's
Yeah.
not coming from, the often billions of dollars the university has.
No, absolutely. It's coming from, I mean, you get a startup. So at UCLA, I had like a generous startup that allowed me to kind of pay people until we got grants, but then it's funded by NIH grants or whatever grants you're getting. I had NIH grants. And that is essentially what you're using to pay everyone and all of the research that you're doing in your lab, including things like, microscope, uh, maintenance and all these sorts of things, which is something we don't pay for here either.
Mm, interesting. So just out of curiosity, so like culturally, how is Australia different from the US? I've never been there. In some ways, obviously, we're both English speaking. There's going to be some similarities, but what are some of the biggest differences?
Yeah, I think, I mean, I was really lucky to have the colleagues I had at UCLA. I feel like they were super supportive in my area. So in behavioral neuroscience, I had a lot of really close colleagues that helped me a lot, and I helped them a lot and things like that. Uh, but there is generally a more competitive environment in the US within departments, I think, and there's more kind of outward competition for things like space and resources and things like that, which I think is the same at many US institutions, not just UCLA. At Sydney, it's a little different. We tend to share things a little bit more, and space is like a little bit more flexible. So if you need that space, you have it. If you don't, your colleagues might be able to use it, all those sorts of things. So it's a little more fluid. And I feel like the department as a whole is more of a team. We're a team that's trying to, be a great department and make sure people in our department are supported and happy and doing the best research they can. And we will, give what we can to help do that. And I think that is different from the kind of more competitive research institutions in the US.
hmm And just outside of the lab, outside of your professional life, how is life different in Sydney compared to, say, a major U.S. city, just as a person?
Yeah, sure. So one thing that's really interesting, no one emails me outside of 9 to 5. So it is expected you are off. And I tended to be like that in the US anyway, but people will email you at any time of day. They'll email you Thanksgiving, they'll email you Christmas. It's not like here, it's kind of frowned upon. You're like not supposed to email people out of hours. And if you do, you want to have something that tells them that they don't need to respond out of hours. And so it does feel like a better balance of life here. I walk to work. In LA, obviously, I was driving to work. I was in the car for probably 2 hours a day,
Yeah.
if not more. And here I walk to work and I walk home, and I spend a lot of time with colleagues and friends outside of work. So we tend to all get together on a Friday night as colleagues, and that's like a very Australian thing. And I think that's a nice way to kind of solidify, your friendships and relationships, and spend a lot of time with family, which we didn't have in Los Angeles. I think it's something that you don't think about when you're going to get your big job. You just want to get the best job that you can, but you don't necessarily think about who's going to be there and who your family and your community are going to be that allow you to live a happy life.
hmm Um, are there any final thoughts or things you want to reiterate about what we talked about today with respect to dopamine and addiction, the hypothalamus, and all that stuff?
I guess I'd just say I think everything's more complicated than what we think it is. And I think we're going to be continually trying to understand the complexities of the brain, and that, we should not think about them as being too simple.
All right, and it sounds like you've got a paper or two coming out. Can you remind us what those are about and when we might see those?
Yeah, so we just had a paper last month out in Nature Neuroscience, and we were trying to understand how dopamine might be encoding value. And we found evidence that it's not at a learning relevant frequency, but at higher frequencies, it seems to be signaling value, and it is valuable to the animal. And this might suggest a dissociation in what dopamine's able to do in learning settings relative to, for example, drug settings or something like that. We just had a paper accepted last week in Journal of Neuroscience, and what that's going to be looking at is how the dopamine release in the nucleus accumbens core is signaling the general excitatory components of learning, and this is actually not capturing a reward prediction error. So we see something that's really interesting there that doesn't seem to be along the lines of canonically what we've thought that signal is doing. And again, it's this really specific learning signal. It's not just a reward prediction error. It's doing something unique from other areas of the nucleus accumbens probably, and also striatum.
Interesting. Well, thank you for your time. This has been fascinating. I used to think about the hypothalamus a lot, and I haven't thought about the lateral hypothalamus in a while, but it's an interesting area. Melissa Sharpe, thank you for your time, and I hope to talk to you again in the future.
All right. Thanks, Nick. Take care.
This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.